Hi, today I wanted to talk to you about cellular respiration and fermentation. These are both types of energy conversion within a cell. So first let's talk about cellular respiration. Cellular respiration is an aerobic process. That means it requires oxygen. It's an aerobic process used by cells to break down glucose and release energy to form ATP. And we'll talk about what ATP is. There's an equation for cellular respiration. Equations have addition signs or subtraction signs in them. And so this is an equation, not a formula. This portion here is a formula. This portion here is a formula. But this entire thing is an equation. This is the equation for cellular respiration. So this equation reads 1 glucose plus 6 oxygen yields 6 carbon dioxide and 6 waters. So glucose in the presence of oxygen can be broken down by a mitochondria or by cellular respiration to form CO2 and water. Now you'll notice there's no ATP written in this equation because this represents just the atoms. So ATP is formed when the energy is released, when that glucose is broken apart. So cellular respiration involves 3 processes. It involves glycolysis, Krebs cycle, and electron transport chain in that order. So let's talk about what ATP is before we go much further. So ATP is an energy molecule that's used by all cells. ATP stands for adenosine triphosphate. So this whole part over here, this ribose and adenine together make up the adenosine, and this part over here is the triphosphate. The reason why we know that is because tri means 3, and we have 3 phosphates here. Now what happens is when the cell needs energy, what it does is what it will do is it will break one of these phosphates off of this adenosine triphosphate, and we will get adenosine diphosphate. And it's adenosine diphosphate because it has 2 phosphate groups. And when that happens, energy is released, and that energy can be used by the cell. So adenosine triphosphate is like having a full battery, a full charge on your battery. Adenosine diphosphate is like having a battery that's running out of energy. So we can break apart glucose, and we can make a whole bunch of ATP from ADP. So this arrow can go in both directions. In the direction you see it here, this is actually releasing energy. If we were to flip it the other way, then the molecule would be absorbing energy. This is a mitochondria. This is an actual photograph of a mitochondria. And what we're going to do is we're going to actually draw this molecule, or this organelle. This organelle is used for cellular respiration. So what I'm going to ask you to do is I'm going to ask you to draw an oval, or a kind of a peanut shape like this. And then inside I want you to draw a second line that's a squiggly line just like this, that's closed. And you can see we've got several different parts. In this drawing, we're pointing to the outer membrane. So on your drawing, the outer membrane will be this outer line. And you should probably make this fairly big because in future slides, we can actually add information on this picture. So if you make this picture about a half a page, that would be fantastic. So the outer membrane is this outer boundary. So the outer membrane is the outermost line on this mitochondria. Then there's also the inner membrane. On your drawing, it would be this squiggly line in the middle. That is the inner membrane. And on this particular one, you can see we've got an inner membrane that comes across here, and then it goes back. So there's a little bit of space in between there, but this line right there, that is the inner membrane. So we have the outer membrane and the inner membrane. Then between those two, we have what's called the intermembrane space. Intermembrane is one word, and you can think of intermembrane as between. The word between and inter both have T's in them. So the intermembrane space is this space right here between the membranes. So it's inside the outer membrane, outside of the inner membrane. And then last but not least, we have the part called the matrix. The matrix is this innermost part. The matrix is the portion within the inner membrane. There's also one more term. It's called cristae. It's C-R-I-S-T-A-E. And cristae are literally these folds. So they're literally the folds in the mitochondrial inner membrane. So cristae, C-R-I-S-T-A-E, cristae are literally the folds within the inner membrane of the mitochondria. All right, so glycolysis. Let's talk about the three parts of cellular respiration. Glycolysis is the first. Glycolysis actually occurs in the cytoplasm, outside of the mitochondria. In order to take a glucose molecule and break it in half, which is what glycolysis does, we need 2 ATP. And you can think of it as one ATP is going to grab onto one side, the other ATP is going to grab onto the other side, and it's going to break this glucose molecule in half. So we have to put in 2 ATP to get this process started. Glucose has 6 carbons, and that's what each of these gray spheres represents. And what we literally do is break this in half, and you put in 2 ATP to get this process started. What happens is we take one glucose molecule and we break it in half, and the results are 2 pyruvic acids, also known as pyruvate molecules. So we take this one glucose, we break it in half into 2 pyruvates or pyruvic acids. In the process, when we break this in half, breaking this bond releases energy, which is great because we invested 2 ATP, and in the process, we got 4 back out. Now, in this picture, it's only showing that 2 ATP are made, but it cost us 2, and we made 4. So this picture is showing the net gain of ATP. There's actually a net gain of 2 ATP molecules. So it takes 2 molecules to get it started, but we get 4 back. So we profited 2. Pretty good deal. We also get something called NADH. NADH comes from NAD+. NAD+ is an electron carrier. And what happens is NAD+ will pick up 2 electrons. It'll also pick up 2 hydrogens. And, it'll also pick up 1 hydrogen. And it becomes NADH. And NADH is then used for a later process. The 2 ATP that are made here are used for whatever the cell needs to use it for. So this process is called anaerobic. It's called anaerobic because it does not require oxygen. The second part of cellular respiration is the Krebs cycle. And the Krebs cycle happens in the matrix of the mitochondria. So you may want to make a note on that picture that you drew. You may want to write Krebs cycle next to the matrix, or next to where you wrote the word matrix. So Krebs cycle is the process, the location is the matrix. The Krebs cycle requires oxygen, so it's considered an aerobic reaction. You can think of it like doing your aerobic exercises. It's aerobic because it requires oxygen. It does not use oxygen directly, but the electron transport chain does. And if the electron transport chain backs up, the Krebs cycle backs up. Therefore, the Krebs cycle is said to require oxygen. So the Krebs cycle turns twice per each glucose molecule. That's because there are 2 pyruvates or pyruvic acid molecules. We need to send both of those through the Krebs cycle. Each turn of the Krebs cycle, each time we send a pyruvate or pyruvic acid completely through this cycle, we get NADHs. So for each turn, we get 3 NADH molecules. And we're going to put pyruvic acid, both pyruvic acids through. So we have to multiply that by 2 to get the amount per glucose. So we get 6 NADHs. And those are high energy molecules. They're carrying electrons that are going to be used in the next process. We also get one fad H. A fad H, I joke around saying fad is so fabulous. There's only one of them per each pyruvic acid. But there are 2 pyruvic acids for each glucose that we started out with, so we get 2 fad H2s. We also get CO2. You remember glucose starts out with 6, right? Glucose starts out with 6 carbons. What happens is when we break it apart, the pyruvates or pyruvic acids each have 3. So each of those carbons, as we go through the Krebs cycle, each one of those becomes a CO2. Each one of these in the other pyruvate becomes a CO2. So for each turn of the Krebs cycle, we get 3 CO2s. When we send both pyruvates through, or both pyruvic acids for the one glucose that we started with, we get 6 CO2. Now, the electron transport chain and ATP synthase is where we get all of our ATP from, or the vast majority. So far, we've only gotten 2 ATP from from glycolysis. We've gotten 2 from the Krebs cycle. That's not very much. 4 ATP is just not enough. So what's happening is we now have these NADs and these FADs, NADH and FADH2, that have these high energy electrons. And we need to get the energy out of those high energy electrons. That happens at the electron transport chain. The electron transport chain and ATP synthase are part of the inner membrane of the mitochondria. The NADH and the FADH2 carry their electrons and their hydrogen ions to this location. They actually bring their electrons and their hydrogens to the electron transport chain. So let's review. In glycolysis, we had 2 NADHs made, and in Krebs cycle, we had 8 made. Now, we're going to ignore this last column for just a moment. In glycolysis, we didn't make any FADs. Remember, they're so fabulous. We only get 1 at a time, one per pyruvic acid. So we get a total of 2. Well, each NAD has enough energy to make ATP, and FADs have enough energy to make ATP. So if we have, for every NAD that we have, we can make 3 ATP. So if we take 8 plus 2, we have 10 NADs. Each NAD can give us 3 ATP, so we can get a total of 30 ATP from those NADs, and I'll show you how in just a minute. So FADH2, we have 2 of those from the Krebs cycle. Remember, one from one pyruvate and the other one from the other pyruvate. Each one of those FADs has enough energy to make 2 ATP. So if we take 2 times 2, we get 4. So in the process of the electron transport chain, we can make 34 ATP. That's pretty darn good compared to the 2 that we got in the first process of glycolysis and the 2 that we got during the Krebs cycle. This will have a much better yield. We'll get a total of 34 ATP from this process. The electron transport chain is aerobic. In other words, it requires oxygen. And oxygen, you need to know this, oxygen is the final electron acceptor. So what's happened is we've taken electrons out of the glucose. NADH and FADH2 have picked them up. Now they're going to send them through a process in the electron transport chain, and someone needs to pick them up at the end. And that molecule is oxygen. Oxygen is the final electron acceptor. So let's take a look at a picture of this. You can see this is the cell membrane, because here's a bunch of phospholipids. Here's a bunch more phospholipids and more phospholipids, and here as well. We've got these large proteins that are embedded in the membrane of the mitochondria. And what happens is NADH and FADH2. FADH2 is not in this picture, but FADH2 does the same thing that NADH does. They bring the electrons and they bring their hydrogens. So they drop off their hydrogens here, and their hydrogens are pushed out to this outer area. So they bring them into the matrix and they're pushed into the intermembrane space. And you can see your labels here so you know where we are. So the NADH and the FADH2s come into the matrix. They drop off the electron, and the electron is passed like a hot potato from one protein to the next, all the way down the electron transport chain. And this is actually drawn literally with this line descending. The reason why is because as this electron is passed from one to the next protein, it gives off a little bit of energy. That energy is what is used to pump the hydrogen ions from the matrix to the intermembrane space. Well, when you're pumping all those through, you're going to have a very high concentration of hydrogen ions in the intermembrane space and a low concentration in the matrix, and that will cause an effect. We'll explain that in just a minute. But let's talk about this electron. We keep passing it from one molecule to another, one protein to another. Now we're at the end. Somebody's got to take responsibility for that electron. They're dangerous just being out there without being attached to a molecule. This is why we breathe in, because what happens is we take an O2 molecule, we cut it in half, and we add 2 hydrogens to it, and we get H2O. And that oxygen accepts the electrons, binds with 2 hydrogens, and becomes water. So yes, your body actually makes water. So oxygen is the final electron acceptor, and then water is formed. So let's talk about, that's what happens to the electrons. Let's talk about what happens to the hydrogens. We have a bunch of hydrogens that have been pushed into the intermembrane space. So we have a whole bunch of hydrogens here, and they're going, oh, I don't want to be here. They want to go from an area of high concentration to an area of low concentration. Well, guess what? There's a protein right here that's very much like a tube. And what happens is these hydrogens go nanner, nanner, nanner. I can go back. And what they do is they flow through this tube-like structure. This tube-like structure is called ATP synthase. Every time a hydrogen molecule flows through ATP synthase, it causes ATP synthase to spin. And if you've ever seen turbines at a dam, you go to Hoover Dam, if you ever get to go, it's a really cool place. There are turbines that the water causes to spin. The spinning motion gives off energy. Well, ADP, I told you we'd be talking about that. ADP picks up that energy and attaches another phosphate and becomes ATP. So we go from ADP to ATP. Now, if we break apart the name of this molecule, it actually makes sense. ACE is an enzyme. The ACE ending means enzyme. To synthesize something, or synth, means to make. So it's an enzyme that makes. And the first part of the name is ATP. So it's an enzyme that is used to make ATP, and we can see here how that process is happening. This is the electron transport chain and ATP synthase. This is how we get those 34 ATP molecules, and this is where the bulk of the ATP molecules are made. So if we put all of these together, you can see we've got glycolysis in the cytoplasm, and it takes glucose and breaks it into 2 pyruvic acids. When it does that, 2 ATP, a net of 2 ATP are made, and we also get 2 NADHs from that process. Now this dashed line is here because everything after this process we have to do each time with each pyruvate. So what happens is we send 1 pyruvate through, and when we do, we get 1 CO2, and we multiply it by 2 because we have two pyruvates. That pyruvic acid goes into the Krebs cycle, which happens, or excuse me, this is the process that happens in the matrix. The matrix is the location. When the pyruvic acid goes into the Krebs cycle, it goes through one rotation. In that one rotation, 2 ATP are made, 2 CO2 molecules are released, times 2. Then we've got NADH that are made, 4 of them are made, times 2, because there's a second pyruvate coming through. And 1 FADH2 is made, times 2, because we have another pyruvic acid that's coming through. These NADHs and FADHs all go to the electron transport chain. The electron transport chain is on the inner mitochondrial membrane, and that's where we get the vast majority of our ATP. We get between 32 and 34 ATP. So if I were to ask you how many ATP are produced by cellular respiration, you should take these numbers and add them together. So we've got 2, 32 to 34, 2, 32 to 34. So we've got between 36 and 38 total ATP from cellular respiration. So here's a summary. I would strongly recommend that you pause the video and you copy down this data table, because this is a nice, neat way of representing where each process takes place. Tells you how many ATP are made in each process, how many NADs and how many FADH2s are made in each process. It's very helpful, and this is the bulk of what you would need to know what you would be tested on. So I would pause the video now. All right, so here's another way to summarize it, and I'll let you take a look at this if you'd like to pause the video. All right, fermentation. Fermentation is much more simple than cellular respiration. So fermentation occurs when there's no oxygen present. Fermentation is an anaerobic process. In other words, no oxygen. There are two types of fermentation. There's lactic acid fermentation, and this happens in your muscles. This is what can make your muscles tired and burn. It's that lactic acid production. There's another type of fermentation which is called alcoholic fermentation, and this actually happens in yeast. This is how we make wines and beer and things like that, and ethanol is actually produced. The difficult thing about fermentation is we only get 2 ATP. That's because we go through glycolysis and then we have to regenerate that NAD in order to break glucose apart again. So there's only 2 ATP versus the 36 to 38 that are made in cellular respiration. Big difference. So let's look at these two different types. In lactic acid fermentation, we start with glucose. We go through glycolysis, just like in cellular respiration. We get 2 pyruvates. We go through the rest of fermentation and we get 2 lactates. The part of this second part, the reason why this second part actually happens is because we need to free up the NAD+. NAD+ picked up 2 electrons and hydrogens, and now it's holding them and it goes, I can't take this stuff. You can't break another glucose because my hands are full. So what happens is we have to free up those electrons, or excuse me, free up the NAD+ by getting rid of those electrons and getting rid of the hydrogens. So what happens is NADH will give its electrons back to the pyruvates and give the hydrogens to the pyruvates, forming lactate. And what happens is we get those NAD back so we can break another glucose. The same idea happens in alcohol fermentation. The big difference is in alcohol fermentation, we also have CO2 that's produced. So you can think of alcohol as, if you think about beer and champagne, they're bubbly. That's because of the CO2. The reason why some wines are not is because in the process of the fermentation, they allow the bubbles to escape when they're creating wine at the wineries. So same thing happens here. We need to free up that NAD plus again. So NADH is going to give its electrons and its hydrogens back to the pyruvate, and that pyruvate is going to become ethanol. So these two processes differ slightly, and the reason why we have two different processes is because different organisms do these. We do lactic acid fermentation. Yeast does alcohol fermentation. And that's what I have for you today. It's a little bit of a long video, but it's chocked full of good information. And if you have any questions, come see me in class.